Hello, I am Fran Yannick. I am Dr. Bruce Cohen's clinical nurse at Akron Children's Hospital. When I was asked to introduce Dr. Cohen to a UMDF audience, I wondered what I should say about a person well known to most of you because of his dedication to the UMDF for the past 25 years. Wow, I could list the credentials which would take all of my time and can be found quickly on the internet. But my time working for Dr. Cohen and as the parent of one of his patients, this is a story that parallels the history of the UMDF and the journey many of you have taken. I was working at the Cleveland Clinic as an RN in cardiac research in 1998 and had been looking for a neurologist for my daughter, who at age 33 had carried a number of diagnoses including cerebral palsy, lupus, and a coma of unknown origin, depending on which doctor she was seeing. Dr. Cohen was suggested to me because he was starting to be known for his expertise with hard-to-diagnose patients. At that time, I also saw that Dr. Cohen was presenting something called Mitochondrial Disease at our Thursday medical grand rounds, so I attended to see who he was. As he described a very new area of medicine, so new that most doctors knew little to nothing about it, and a few disbelieved altogether, I was convinced he was the one my daughter needed to see. And now, 20-some years later, I know I made the right decision. His knowledge, research, care, and relationship to Chris and our family has enabled Chris at the age of 55 to be a happy, engaged adult who is still Dr. Cohen's patient. Many of you know the relief of finding a doctor who understands this complicated world of the difficult-to-diagnose patient. When Dr. Cohen was looking for a nurse, I applied for the position in pediatric neurology because he felt my personal knowledge of mitochondrial disease would be helpful. And so began our journey in the world of mitochondrial medicine, where the muscle biopsy was the current means of a diagnosis, and genetics was far in the future. 10 years ago, Dr. Cohen became Director of Neurology in the Neurodevelopmental Science Center at Akron Children's Hospital. I was happy to join him there, and he was soon appointed Director of the entire Neurodevelopmental Science Center and was recently named Interim Vice President and Medical Director of the Rebecca D. Considine Research Institute, where he is doing exciting things we only dreamed of 25 years ago. We are now opening the doors to a mitochondrial center of excellence at Akron Children's Hospital, which will enable us to evaluate and treat many more patients looking for a diagnosis. It has been my honor to work with Dr. Cohen, and it is my pleasure to introduce him to you.
Good day, everybody. It's my pleasure to come talk to you today about mitochondrial diseases. My name is Bruce Cohen, and in celebration of our 25th year, I'm really excited to be here to discuss this topic with you. My first mitochondrial meeting was in 1997, and I've been to pretty much every UMDF meeting since that time. So let's just hop right in. The mitochondrion is depicted in this picture, showing the general structure of what a mitochondria would look like. It's obviously going to be a little more complicated than this, but this is not a bad place to start. And the mitochondria is to the cell and is to the body what a car engine is to the car. And the purpose of the mitochondria, simply put, is to make a rechargeable source of energy. And we're going to be talking about what that is, and that's called ATP. And that happens on the walls of the mitochondria. So this is what a molecule of ATP looks like. ATP is short for adenosine triphosphate. In this picture, there are three main parts of the molecule. There's the adenine, and there's the ribose, which is a 5-carbon sugar, and there's 1, 2, 3 phosphate groups. The adenine and ribose forms a compound called adenosine, and then triphosphate 1, 2, 3 phosphate groups. The energy within this molecule is much like a charged battery, and it exists right here between that second and third phosphate group. And although it's pretty difficult to understand how this actually holds energy. It does. And the energy is like a loaded spring right here. And the body can use ATP to do work by releasing the energy within this spring. It releases it with water and causes an energy reaction to take place. And the purpose of the mitochondria is to actually reload that phosphate group, almost like pushing a spring till it clicks, and where it becomes activated again. So adenosine triphosphate is the charge battery, and when it releases the phosphate group, that's the discharge battery. In our bodies, adults have about 17 grams of ATP in their body. 17 grams is a small fistful of almonds. So that gives you an example, an idea of how much or how little ATP there is actually in the body. Now, ATP, as I said on the previous slide, has to be recycled. And we're just going to start up above. Here's the ATP. And again, that's the loaded charged battery per se. By releasing the phosphate group, it releases energy for the cells to use. That's energy for the eyes to see, the muscles to contract, the heart to beat, the ears to hear, the kidneys to make urine, and so forth and so on. When that phosphate group is released, we have a phosphate group and adenosine diphosphate too, and this is a discharge. The body really can't do anything with it. It has no more energy. And the purpose of the mitochondria is to take the energy we have from food that we eat, add that phosphate group to recharge the battery to make ATP again. So the mitochondria recycles that 17 grams, which is about a half an ounce of ATP, into 95 pounds of ATP each day. It's just an unbelievable ratio that our body and our mitochondria are able to recharge that little bit of ATP that we have to make 95 pounds. Now, our body uses those 95 pounds of ATP every day. So how many ATPs are we really making? It's an unbelievable amount of ATPs. I'm just going to show you the process of complexes 1, 3, 4, and over here is complex 5. Every time that wheel spins, it's charging up another ADP to make ATP. In our bodies right now, each ATP is making 6 molecules. I'm sorry, each complex 5 is making 6 molecules of ATP a second. So as we sit here as an adult, you're making 530 quintillion ATPs every second, and that means that you're using 530 quintillion molecules every second. You can actually make more if you're doing work. So if you're sitting here, that's how much you're making and utilizing. If you get up and walk around, double that. So that's actually a half a gram of ATP per second, which is the amount in a small raisin. So our bodies rely on that small raisin size amount of ATP every second for the energy. And just imagine a raisin every second as we sit there of ATP. If your mitochondria aren't working right, they're not going to make enough ATP. And that's when you start getting into energy failure. And that's essentially what a mitochondrial disease is. The mitochondria not making enough ATP to meet the needs for the energy requirements of all the organs of our body. So in terms of how much energy we're making, I'd like to give you some examples because it really is all about the energy. Our brains use about 11 watts of energy, which is about the equivalent of 2 night lights worth of energy. That's what it takes to run our brain, 11 watts. And that's second after second after second after second. As we sit there, our entire body is using 75 watts of energy. And I've depicted that as a light bulb that we would have in our kitchen, lighting our kitchen, these 75 watt floodlights. So just sitting there, that's what our bodies utilize. Now, a trained athlete can increase that energy production and energy utilization tenfold. So that would be 750 watts of energy for a trained athlete. Obviously, people can't, even a trained athlete can't do that for very long. Again, our mitochondria can upregulate the amount of ATP it makes, but only in a trained athlete. An untrained athlete, a weekend athlete, 5 or 6-fold increase. So really, we spend our day sitting around. We're generating or utilizing about 75 watts of energy to 150 watts of energy on a continuous basis, depending if we're sitting around or walking. Walking briskly, 3 times at 75. So what does a car do? A Tesla at rest, when the car is activated but nothing is running, that Tesla runs off of 25 watts, but can at 60 miles an hour utilize 16,500 watts. And the reason I'm going through all these examples is that what happens if that car cannot generate 16,500 watts of energy? It's not going to make it to 60 miles an hour. And although a car can run perfectly fine at 30 miles an hour, it's not going to do the job of what your car wants to do. Likewise, if your mitochondria aren't putting out the proper amount of ATP, again, as I said, the brain is not going to function properly, or the eyes aren't going to function properly, so forth and so on. So I'm going to go back to the car analogy. We're going to move from an electric car to a gasoline car, because it ends up that the fuel that a car engine utilizes, octane, is not that dissimilar to the fuel that our bodies utilize in the form of glucose. So this is a molecule of octane. It's 8 carbon atoms surrounded by a whole bunch of hydrogens. And we pump that fuel into our gas tank and we add oxygen. That's what a carburetor would do, or a fuel injector would do, mix the oxygen in the octane. And you put it into the car engine. And what happens when you put gasoline and oxygen in the car engine? Well, if there's no heat involved, nothing. It'll sit there forever because it's at room temperature. The chemicals don't react. But if we add a spark, we all know from our experience that gasoline will explode and create and release energy. Now, what happens when you rearrange the molecular structure of octane, which is carbon and hydrogen, and oxygen, which is oxygen atoms, we create water. Some of the hydrogens come off that octane and are added to the oxygen to make water, H2O. We make carbon dioxide, again, a mixture of the carbon and the oxygen. The explosion takes place, which creates a twisting motion inside the cylinder. An explosion of the piston inside the cylinder creates a twisting motion that's called torque. That's the work that runs the car. We also know that it generates heat, and we also know that not all the octane is fully burned. Some of it escapes as partially burned fuel, and that's smog. And that's how gasoline engines work. Obviously, if the engine can't produce enough energy because something's wrong with it, the car's not going to do everything a car's supposed to do. Our bodies use, this is a molecule of sugar as a fuel. We also use proteins and fats, and they're not entirely dissimilar to this molecule of sugar. It's carbon surrounded by oxygens and hydrogens, so it's a little bit different than octane, but conceptually it's the same thing. And we breathe in oxygen, from the air, and our engine is our mitochondria. And our spark plugs are the vitamins and cofactors such as coenzyme Q10, lipoic acid, the B vitamins, folic acid. There's actually a metal core in complexes 1 and 3 called an iron sulfur core. In complex 4, we have a copper core. So there's actually an electrical circuit that goes on, and the explosion takes place. What's produced is the same water that a car engine would produce, the same carbon dioxide that we exhale, and the explosive force generates that third phosphate group onto the ADP molecule to make ATP. This produces heat, not dissimilar to the heat of a car engine. This heat warms our bodies, and it's also not completely efficient. Some of the unburned fuel escapes as lactic acid, as well as some of the oxygen picks up an extra electron and creates free radicals. So what happens when the mitochondria don't make enough energy, don't function properly, not enough energy is produced? Excess free radicals form, which further injure the mitochondria and the cells. The cells don't function properly, and the organs don't function properly. So this is the classic mitochondrial phenotype. Phenotype means what does the person with the disease look like? And this mitochondrial phenotype was put together over time back in the 1970s and 1980s. And as it ends up, the physicians that first described mitochondrial disease in humans really got it right. Um, as most of you know, mitochondrial disease can affect multiple organ systems. It typically does, but in patterns that differ from mitochondrial disease to mitochondrial disease. And we're going to go over a few of the classic mitochondrial diseases in a little bit. But someone with a mitochondrial disease does not have all these features, but select features from some of these groups. So I'm going to list all these features. The first being brain, stepwise central nervous system or brain deterioration. This can result in seizures, movement disorders, ataxia, which is imbalance. It can affect the eye muscles, which can create droopy eyelids, and the eyes not moving to the left or right or up or down properly. And that's called progressive external ophthalmoplegia. It can injure the optic nerve, creating optic atrophy and loss of central vision, or a condition affecting the retina, which is a little bit different part of the eye, which causes night blindness. It can affect the ears and cause high frequency hearing loss, which is a big deal because the hard sounds of our speech, like the P's and the D's and the T's and the B's, are the high frequencies. And when you lose that, speech becomes very muffled. The heart has its own electrical conduction system, and people with mitochondrial disease can have conduction defects. The muscles are the most obvious part of the body affected by adult onset mitochondrial disease. Myopathy means muscle weakness. Cardiomyopathy means heart and muscle weakness. The liver can be affected. The nerves can be affected. Actually, the formation of fat can be disrupted as well. So patients can get either loss of all the fat in their body or development of fat accumulation called lipomas is part of their disease. Often there are classic brain MRI findings where the deep parts of the brain are affected, and there'll be one or two images that we're going to show later on. And we can measure some of the abnormal chemicals in the bloodstream that are suggestive that the mitochondria aren't working properly. Lactic acidosis, there's certain amino acids that are elevated or reduced in some mitochondrial diseases, and they're intermediate chemicals in mitochondrial metabolism called organic acids that can be elevated in patterns that are highly suggestive of a mitochondrial disease. So as a physician, when we see one or more of these symptoms in particular combinations, we can start to think about mitochondrial disease. So certainly in someone with high frequency hearing loss, and that's all they have, one wouldn't necessarily think about mitochondrial disease, the same thing as cardiac conduction defect. But when you start mixing myopathy, muscle weakness, and retinitis pigmentosa, then you start thinking, well, maybe could this patient have a mitochondrial disease? And you mix any of them with droopy eyelids and progressive external ophthalmoplegia, then certainly the chance of it being a mitochondrial disease rises considerably. So let's talk about the classic description of a mitochondrial disease, Lee syndrome. Lee syndrome was first described in 1951 by Dr. Lee. And this is a disorder often affecting young children with progressive dystonia, which is a twisting movement of the limbs. Often these patients have optic atrophy. Often these patients have seizures. They can have respiratory depression because the brain gets injured in a way that doesn't allow them to breathe properly. This is a chemical description of the molecule lactic acid. And the brain we talked about is having deep gray matter injury. This white area of the brain, in what's section of the brain is called the midbrain, is one pattern of injury that can be seen in Lee syndrome. So again, Lee syndrome, interestingly, primarily affects the central nervous system, that's the brain. And as we talked about, control of movement, loss of cognitive function, but not always, epilepsy, but not always, loss of respiratory drive, normal breathing patterns, but not always, swallowing difficulties, almost always. Um, but also on occasion, depending on what the cause of the Lee syndrome is, it can affect the liver, the heart, the muscles, the kidneys, or even other organs. And Lee syndrome is not one disease. There's over 75 different genes that are involved, that mutations in those genes that can lead to Lee syndrome. We don't know what the common final pathway is that why one gene will cause Lee syndrome and mutations in another mitochondrial gene, just as important, causes a completely different syndrome. Primarily, the onset of Lee syndrome is between 6 months and 5 years of age, but it can occur earlier or much, much later. And it's generally a progressive disease, often marked by long periods of disease inactivity. So the patient will have their first Lee syndrome presentation and then could be stable for several months or a year before the next progression of the disease. We have patients that can go 5 or 10 years or longer between progression of the disease. So there's still a lot we don't know about Lee syndrome, and actually the UMDF and other international patient advocacy groups have made this a priority of investigation and put a lot of money into research regarding the natural history and experimental therapeutics for those with Lee syndrome. The next description is Milos, that stands for mitochondrial epilepsy, lactic acidosis, and stroke-like syndrome. And this was a disorder that was well described in the mid-1980s. And these patients, generally presenting in adolescence and young adulthood, would present with strokes and epilepsy. High-frequency hearing loss is common. Diabetes is common, and muscle weakness is common. This is an MRI scan picture showing the multiple strokes. Again, the area of injury is appearing in the whiter shades of gray. This is a large stroke, but these are small strokes that have occurred as well in patients with Milos. Now, Milos, again, is even more highly variable than Lee syndrome. There are people with a common Milos mutation that all they have is high frequency hearing loss and diabetes. There are other patients with Milos syndrome that have myopathy or strokes and not much else. So it's really a very highly variable syndrome. The most common mutation, or the frequent mutation for Lee syndrome, is this mutation in mitochondrial DNA known as the A3243G mutation. It's not an uncommon mutation. About 1 in 1,000 people have the mutation, but in low enough heteroplasmy that only 1 in 5,000 people actually have the disease. I'm going to move on to another syndrome called Alpers-Huntenlacher syndrome, or Pol-G spectrum disease. Again, this was first described as a fatal infantile disorder in 1931 by Bernard Alpers, the man on the top. And it took till 1976 before Peter Huntenlacher described the liver aspects of the disease. So again, initially it was thought to be primarily or only a brain disease of horrible epilepsy, developmental regression, and cortical blindness affecting youngsters. But as treatment for epilepsy improved, some patients lived longer and longer, and if they lived long enough, they often got liver disease, and that's what Peter Huntenlacher described. There are two physicians, scientists, Dr. Bill Copeland on the left and Bob Navio on the right, that working in different parts of the country, but collaborating really in the 1990s and early 2000s. These are members of the United Mitochondrial Disease Foundation, have done a lot of work for the UMDF, really puts whole G spectrum disease on the map. Dr. Copeland characterized and cloned the human polymerase gamma gene. That's the protein that replicates mitochondrial DNA. And Dr. Nabio added clinical context to this, and after a decade of work showed that mutations in this gene can create a spectrum of illness, including Alper's disease. And that was really, both of their discoveries were highly impactful, settled really decades-long argument about this. Now, this is just a map of the different mutations in this one gene that can cause human disease. As you can see, there are about 150 different mutations on this one gene. Each of them is potentially responsible for causing a number of different variants of whole G spectrum disease. In 2005, this could only be done in research laboratories. By 2008, the clinical laboratories had picked up the ability to sequence the gene. And now at a fraction of the cost of what we paid for it, sequencing in 2008, this gene is routinely sequenced, and we pick up a lot of patients with this disease, again, with advances in molecular diagnosis. When we talk about whole exome sequencing and mitochondrial DNA sequencing, it's done in a machine that looks like this, not much bigger than our home computer. And within 2 or 3 days, we can have the whole human genome sequenced. We used to put patients through muscle biopsies as the method of diagnosis. This is a ragged red fiber, and ragged red fibers are one of the quintessential features of some, but not all, mitochondrial diseases. Here's another ragged red fiber. We tend not to do many muscle biopsies anymore because gene sequencing has a higher yield and is probably, in real terms, about 1/50th the cost of putting a patient through a muscle biopsy. I did much of my work with Dr. Hoppel earlier in my career. We did a lot of muscle biopsies. His laboratory ran the specimens. I provided him with the muscle tissue to run the specimens, and we did enzyme analysis, electron transport chain enzyme analysis. And although this is still done, it's done less frequently. We get most of our diagnoses with the genetic testing. When we talk about treatment for mitochondrial diseases, and again, we'll hear more about this in other talks, we used to prescribe lots of vitamins. I've never prescribed this many vitamins. This is one of my patients that came in who I think was taking so many vitamins, there was no room in his stomach for food. But we still do prescribe some vitamins for mitochondrial diseases. But luckily, our therapeutics are going to be more finely tuned and aimed at really getting to the root of the problems. And I think really in the next few years, we're going to have some medications that will improve the lives of our patients, and probably in the not too distant future, and actually part of this meeting, we'll be hearing about some gene therapies that will be helpful. We want to cure, and I'm not saying that we should not use the word cure, but or we should use the word cure, but some of these gene therapies really look like they could lead to cures for some people with mitochondrial diseases. So I'm going to end that talk now, my talk now. Again, the mission of the UMDF is to promote research and education for the diagnosis, treatment, and cure of mitochondrial disorders, and to provide support to affected individuals and families. This was a very brief overview of what are mitochondrial disorders. I hope you've learned a little bit from my talk, and look forward to meeting all of you in person next year. Thank you.